Welcome everyone to our R4Bioinfo series, which we started in September 2020. Today, we have the pleasure to have with us Dr. Nickolay Oskolkov. He will present the second part of the workshop, Omics Integration and Supervised Machine Learning. But before introducing our speakers, so we will introduce you to the R Ladies and R Ladies Tunis initiative. Please, Monet, can you share your screen?
So,
hello and welcome to this workshop session. Before starting the workshop, we'll introduce you to the R Ladies Global and R Ladies Tunis initiative. The R community suffers from an underrepresentation of minority genders in every role and area of participation, whether as leaders, package developers, conference speakers, conference participants, educators, or users. As a diversity initiative, the mission of R Ladies is to achieve proportionate representation by encouraging, inspiring, and empowering people of genders currently underrepresented in the R community and to facilitate individual and collective progress worldwide. Gabriela de Caros founded R Ladies on October 1, 2012. R Ladies Global was born and the grant was awarded in September 2016. Since then, R Ladies has grown to 170 chapters in 44 countries and 39,000 members. R Ladies Tunis is part of R Ladies Initiative and was created in May 2020 by women working as data scientists and biostatisticians and bioinformaticians. Our goal is to create an R community in Tunis and empower underrepresented genders in the R community. This is the core team of R Ladies Tunis. Come and join our community on social media. This presentation was made with R Markdown. Thank you for watching this video and enjoy our workshop. Hello and welcome.
you are unmuted, yeah, I think. Yes.
Okay, thank you, sorry. So, as I said, today we have the pleasure to have with us for this second part of the workshop, Omics Integration and Supervised Machine Learning, Dr. Nickolay Oskolkov. I am Hediat Nani, I'm postdoc at Pasteur Institute of Tunis and co-founder of R Ladies Tunis, and I will be hosting this session. So Dr. Nickolay Oskolkov is an analyst doing bioinformatics, data science, and statistical analysis. He has a background in theoretical physics, PhD 2007, and he moved to life sciences in 2012, where he was working in computational biology and biomedicine. He's currently employed as a bioinformatician within the SciLifeLab bioinformatics platform at the National Bioinformatic Infrastructure in Sweden. His range of interest include genomics, transcriptomics, microbiome, ancient DNA, single-cell machine learning, image analysis, and evolutionary science. So thank you, Nickolay, for being with us and for this second part of this amazing workshop. Please, the screen is yours.
Thanks a lot. Thanks a lot, Hedia. Hello, everyone. Thank you very much for joining us. And thank you for joining last time, and thank you very much, dear R Ladies, for inviting. I really appreciate it, and it's a great pleasure to talk to you and see you. So thank you very much, everyone. Yes, so I'm going to continue talking about our. It's basically a short version of our course that we are teaching within SciLifeLab training program. And we are running this course twice a year, usually, and this was, last time it was run in April. And next time, yes, I got a few questions about when it's going to be next time. I think I confused everyone last time. saying that it's going to be in July, and that's true, but it's going to be within ISMB conference. So basically, if you are participating in this conference, and you might want to attend our tutorial. So if you go to the conference web page and select tutorials, then you can find tutorial number 4, which is a practical introduction to multi-omics integration and network analysis. And these are the people who are running this course. Ashfaq Ali and Ruben Feitas are my colleagues from National Bioinformatics Infrastructure Sweden. So if you're participating in this conference, you might want to attend this tutorial. If you're not participating, next time we are going to run our course in September, actually. And the dates are from the 6th to 10th of September. And you can apply. So basically, you can monitor. If you just Google SciLifeLab courses, you can go to the training web page of SciLifeLab, and you can click on upcoming events and courses. Our course is not here yet because it's going to be in September. It's a little bit too early to advertise it. So I think the last event is June 14th. But you might want to keep an eye on this web page, and when the announce is available, you might want to apply and participate in this course. So everyone is very welcome. Even though we, as a National Bioinformatics Infrastructure Sweden, have to prioritize participants from Swedish academic institutions, but we are quite flexible, and usually we accept plenty of people from abroad. So if you're not from Sweden, you still might be accepted. So please, you're very welcome to apply. Yeah, so I think we should start. So this is a course schedule. You will find all the course material here if you just click on the links, these hyperlinks. Those are all the slides and are marked down.
Sorry, can you make your screen a little bit bigger, because I see it, yeah, very small.
Sure. Thank you.
Thank you.
Yes, and please interrupt me if if you by any chance you don't see what I'm showing, or you suspect that I'm sort of think that you see this, but you don't actually see.
Thank you.
So please let me know, because I don't see your faces. Not everyone. So all the notebooks, Jupyter notebooks and R Markdown notebooks, and all the PowerPoint slides and PDF slides, they're available. Genomics are comparable. So those two omics are, I call them manageable, because they don't suffer much from the curse of dimensionality, those two omics. And if you sort of interested in omics integration, you probably know that people from metabolomics, they are the ones who started talking about omics integration, and for a reason. So there is a reason why usually people who do metabolomics and proteomics, they use this concept of data integration. While if you are from genomics side, for example, you probably never heard about omics integration, and I'll explain why. So if you're working with transcriptomics, then your data matrix is a little bit elongated in the P dimension. So here, all these values, they are made up. So what you need to see, what you need to watch here is the sort of the ratio between N and P dimensions. So this transcriptomics data matrix, it's elongated with respect to p dimension. But in here you already have this the curse of dimensionality limit p much greater than n. So you need to be careful. If you have if you're working in this limit, you have to take this into account somehow. You have to reduce dimensions, you have to do regularizations, or you have to use Bayesian statistics. So there are basically three ways how to overcome the curse of dimensionality. But if you happen to do single cell analysis, so people are very happy in single cell research because they don't have the curse of dimensionality problem, because they're really working with hundreds of thousands and even millions of cells. So it's becoming almost a routine to sequence millions of biological cells. And there you actually, it's a luxury problem. You don't have this curse of dimensionality problem. But by transcriptomics here, I mean like bulk RNA sequencing, where typically you don't have hundreds of thousands or millions of samples. So transcriptomics is a little bit more difficult. But if you're working with genomics or mutilation data, there you have, at least for human genome, you might have 30 million genetic variants or 30 million CpG sites. So your data matrix becomes very much elongated in the p direction. And this is sort of, I call those two omics impossible. And indeed, it's very hard and very challenging to use these two omics for data integration. And not only for data integration, I have like conceptual troubles trying to understand how to do robust statistical analysis of this very, very ultra high dimensional data. So this is just a recap of what high dimensional data is, what we were talking a lot about last time. And this is just another illustration of what is high dimensional data. So genomics and metabolomics are ultra high dimensional data. So the greater is the p dimension, the more high dimensional data it is. And this slide I'm not going to explain again, just to remind you that usually in biology, in computational biology, biomedical research, we unfortunately have this problem, p much greater than n. So our data is high dimensional, and we can't sort of proceed with statistical analysis without taking it into consideration, this high dimensionality of the data. Because if you simply ignore it and continue with statistical analysis, you get into problems such as if you're doing linear regression, you can get divergences in this limit, in high dimensional limit. Or if you're doing hypothesis testing, if you do statistical test, you know that it's important to have a good, robust variance estimate. Variance in the data is explicitly taken into account when you're doing statistical tests, such as t-test, for example. And it turns out that in traditional frequency statistics, which is called which is based on maximum likelihood principle, there is no way to estimate variance in unbiased way. So basically, it's known and can be derived that maximum likelihood variance estimator is very biased, especially in high dimensional data. So here, what you see on the left side of this equation is estimator, is a variance estimator, what you can estimate from your modeling. While sigma squared on the right hand side, it's a true variance, and it can be derived that the true variance is related with estimated variance through this factor n minus p. So here you get explicitly you can explicitly see the interplay between n and p, and the greater is p, the more your variance estimator is biased. So when p is much greater than n, I actually don't know what you get. I think Carl Pearson and Roland Fisher, the founders of frequentist statistics, they never thought that people would do hypothesis testing in this limit when p much greater than n. They always assumed n much greater than p, and all the equations, all the formalism was derived for actually this limit. So you need to remember about it, and don't just blindly use PCA or linear regression or t-test without looking at the dimensions of the data. And just a few words about the curse of dimensionality again. Yeah, I'm sorry for talking too much about this, but it's really crucial for it's like the major problem. That's why I'm spending so much time. But I promise I am monitoring time, so I hope I will cover everything I planned today. So basically, what exactly going on in high dimensions? Why you get all these divergences and problems? If you simulate data following normal distribution, for example, and you compute pairwise distances between data points in high dimensions. So you start with 2 dimensions, 32 dimensions and 512 dimensions, and you plot what you plot here. It's a histogram of pairwise distances between data points for different dimensionalities. 2 dimensions, 32 and 512, the blue histogram. What you see here is that when you increase dimensionality of the data, your histogram, your distribution is moving to the right, moving towards larger pairwise distances. And the distribution becomes more and more narrow. So what it means is that in high dimensions, distances between data points becoming greater and greater, and more similar to each other. And if you also on this plot, it's also shown that if you plot the distance, average pairwise distance for high dimensional data, and if you now plot a distance between any given point and its first neighbor, middle neighbor, and furthest neighbor, and you increase the dimensionality. So here on the x-axis, it's a number of dimensions on a log scale. It turns out that for high dimensional data, the distances between first neighbor and furthest neighbor, they are becoming more and more indistinguishable. So meaning that there is no difference between your nearest neighbor and your furthest neighbor. What it means is that in high dimensional space, we can't really separate cases and controls anymore. So all data points becoming similar in high dimensional data. So we can't run clustering, for example. All clustering algorithms, they crash in high dimensional space because you can't really discriminate between sick individuals and healthy individuals, for example, or between good cells and bad cells. Or they all become similar. This is a very strange thing about high dimensional data, but this is exactly what is called the curse of dimensionality. So enough about this. I very briefly remind you that by integration, so the curse of dimensionality is a major problem. By integration, data integration, I showed this slide and I said that basically by integration, we mean ability to detect hidden patterns that were invisible in individual omics. So basically, we sort of all data points here, they belong to 3 classes. And x-axis is a latent variable from omic 1, while y-axis is a latent variable from omic 2. And basically, if you project the data points on the x-axis and on the y-axis, all these three classes, you can't really separate data points from three classes. But if you plot these two omics, omic 1 and omic 2 against each other, you suddenly can linearly separate the three classes. So this is the beauty of integration, that you can sort of put in two layers of information next to each other. You can see patterns. You can see that there is some heterogeneity between data points that was invisible, that was hidden in separate in individual omics. And how I evaluate that integration was successful. So this is like the idea of integration. So we want to discover something new, something that was invisible, and it becomes visible after integration. And an indicator of successful integration is a boost in accuracy of prediction. So basically, we use prediction as a criterion of whether our integration model was successful or not. I showed this slide, but very briefly. So if you have three data types, and you can use each of them to predict whether, or try to train a model that can discriminate between healthy and sick individuals. So you can reach some moderate accuracy using individual data types. But if you put them together, ideally, you would like to see an increase in the accuracy of prediction. This is how we understand that there is some synergy between different data types, and they all work together, and you start seeing more and more clear separation between classes. So this is exactly what is going on when you integrate data, that you suddenly start seeing patterns more clearly when you put all data types next to each other. There are different methods. So here today I'm going to talk about Diablo, which is a linear and supervised omics integration method. There are other ones, and we cover more or less all of them in our course. So if you're curious, you can either contact me or you can attend our course and learn them. But briefly, all integrative methods can be divided into linear and non-linear, and supervised and unsupervised. So when you start your integrative omics project or multi-omics project, you first have to ask how much data I have and whether I have a power to do non-linear analysis or I have to stick to linear analysis. That's the first question. Second question is whether I'm doing supervised or unsupervised analysis. Supervised analysis means that you have a clear readout, you have a clear outcome, a disease, for example, or classes of cells, tumor cells and healthy cells. You know, there is a very clear phenotype of interest. Then you would like to do a supervised integrative analysis. While if you're doing more exploratory analysis, more like descriptive analysis, when you just want to see what is in my data, then you might want to do unsupervised analysis. So basically, you have no idea or no clear hypothesis, and you just want to explore your data and see whether you can detect something unexpected, something that you didn't see or didn't suspect previously. And this was a protocol or like a chart of how we typically do integration. Basically, in machine learning view, again, there are different sort of by integration. Maybe other people might understand something else, but here I'm talking about machine learning oriented omics integration. That's why, as a cornerstone of machine learning. You always split your data set into train and test, and ideally validation. You split it into 3 data sets, but at least you need to split it into 2 at least. So the test data set is used at the very end of your analysis just to evaluate how successfully your model works, whether you see actually the boost in prediction. So basically this boost in prediction that I was talking about, you're supposed to see it on this test data set when you evaluate your model, and suddenly you see that there is an increase in the accuracy of classification accuracy. So the trained data set is used for tuning hyperparameters and parameters of your model. So this is where you actually do the integration. This is where you actually put different omics next to each other. But prior to this, you should do feature pre-selection. So it's not like you don't have to do it, but it's highly recommended to do feature pre-selection to be able to do a more robust statistics, more robust analysis. You need to kind of facilitate your analysis to make integrative model to help integrative model. And so we were talking a lot about feature pre-selection last time. And I said that there are 2 ways of doing feature pre-selection. One is supervised, which I was demonstrating with lasso. When you have a phenotype of interest, disease status, for example, and then you can pre-select features that have something to do with this phenotype of interest. You pre-select the features in each individual omic. Or if you don't have this clear phenotype of interest, if you're doing unsupervised integration, you might want to check the variance across samples and just drop features that do not vary across samples. Those are probably not informative. They just contaminate your analysis, and you should not keep them in the integration part. So once you train your model, you evaluate it on the test data set. So I hope this was a recap of what we covered last time. Sorry for spending so much time on this. I hope this is useful because it's a lot of new concepts, and sometimes it might feel too much, and that's why I'm sort of recapitulating it again.
And Nikolay, so
Yes.
we have some questions. Would you like to now answer them or at
Yes.
the end of the session?
Yes, we can take them.
Okay. So John is asking, is there a rule of thumb regarding P quite less than N? Is it an order of magnitude, several orders of magnitude, or?
Yes, exactly. So indeed, it's at least one order of magnitude. So basically, P if P is 10 times greater than N, then people say P is much greater than N. It's like a typical notation in mathematics. P much greater than N, or N much greater than P, meaning that N, for example, here N is at least 10 times greater than P. So basically, if N is 3 times greater than P, then it's not really N much greater than P. But if it's 10 times, so one order of magnitude, then this is really N much greater than P. This is what I mean by using this notation.
So the other question is, what do you mean by the distances between data points in high dimensional data increase?
Yes, so so basically you have samples, and you can and you measure some biological information about your samples, for example, gene expression. And you can use this gene expression in order to compute Euclidean distance between pairs of your samples. basically, yeah, I'm not going into sort of what Euclidean distance is, but basically there is a way to use this gene expression information to establish a similarity between samples, to see how much two samples are similar in terms of gene expression or dissimilar. For example, using Euclidean distance, there are plenty of distances. You can select the one that fits your data types. So once you computed this similarity between samples in pairwise manner, so you compute similarities between all pairs of data points, all pairs of samples, then you can use these similarities and plot a histogram of similarities. So imagine that you have huge vector of similarities between all pairs of your samples. Now you can plot a histogram of these similarities. And this is what is plotted here. So the x-axis here is a distance or similarities between samples calculated from gene expression. For example, gene expression, it can be any other omic. This is sort of density or number of samples with this similarity, pairwise similarity between them. And so what I try to explain here is that your samples become more and more similar to each other in high dimensional space. So that if you increase the p dimension, the more high dimensional data you have, the more similar your samples will look. This unfortunately means that you can't really separate them in a good way. They are so similar that cases look like controls in terms of gene expression. For example, if you're talking about gene expression omic, so you can't cluster in high dimensions. And if you can't cluster, it means you can't train a classifier that can sort of draw a hyperplane between cases and controls in high dimensional space. So they're too similar. They're indistinguishable. I hope this is clear.
Thank you. So the other question is, how do you evaluate unsupervised omics integration?
Yes, so indeed, in unsupervised, indeed, you can't use accuracy. You can't use accuracy as a metric for evaluation unsupervised omics integration. So this sort of framework that I'm explaining will work only for supervised integration. For unsupervised integration, I don't think I have a good answer for you. So basically, by unsupervised integration, I would rather mean finding consistent patterns across omics. So I'm unfortunately not ready to sort of provide a good answer how I would evaluate that integration was successful. So when you do unsupervised analysis, you don't know the ground truth. You don't know what. For example, if you do single cell, it's a typical example of unsupervised analysis. You want to discover heterogeneities between cells. You want to discover cell clusters, but you don't know them a priori. So if you don't know them, you can't really say whether it was a successful integration or not. But you can sort of maybe still using this picture, you can think about unsupervised integration as a way to sort of increase a resolution, increase power of discovering rare cell types. So basically, by putting omics, if I use single cell RNA-seq and single cell proteomics, for example, as an example, by putting those two layers of information next to each other, you might discover a new cell type. This is what I would mean by successful unsupervised integration, discovering a new cluster, discovering a new cell type, which was not visible in individual omics. So if you do t-SNE or PCA on RNA-seq, you don't see this cluster. If you use proteomics, single-cell proteomics, and do t-SNE or PCA or UMAP on this protein level information data, you don't see this cluster. But integrating those two omics, you suddenly start seeing a new cluster, which was not visible in individual omics. This is what I would mean by successful omics integration, but it's not an accuracy. It's not a boost in accuracy.
Okay, so I think there is another question, but we will leave the other question, at the end. But yeah, this one. So what other distance metrics can one use apart from Euclidean Manhattan?
Yes.
So do you have any tutorial exploring comparisons between Euclidean, L2 norm, Bray-Curtis, Mahalanobis, Jacquard, etc.?
Yes, Mahalanobis distance, yeah, Jacquard similarity. Yeah, so basically, it's in different areas, people use different distances because it better suits their particular data distribution. So you're asking a good question. Basically, if you're working with binary data, such as mutilation or maybe open chromatin, ataxic open chromatin information, you might want to choose Hamming distance because this was created for binary data. While if you're working with gene expression, maybe Euclidean distance is good enough for you. If you are working with microbiome, you might want to go to Bray-Curtis distance. So basically, in all omics, people have an idea what kind of distance suits their particular data type. Basically, in UMAP, UMAP has if you're familiar with UMAP, it's a dimension reduction technique. It has a very neat feature, very nice way of integrating different data types in an unsupervised way by providing individual distances for each individual data type that suits this data type. I think that's pretty cool that you can actually specify a distance that fits your data and then integrate different data types.
Okay, thank you. Well, I think we leave the other questions, for the end. Thank you.
Yes. Yeah. So I'm done with sort of recapitulating the previous webinar, yeah. So I just 2 days ago I was listening to a talk, and it was a microbiome research. It was not about integration, but I discovered this kind of this figure that resembled what I'm showing you here. So what I want to say is that here they also sort of using machine learning for microbiome research. They do feature pre-selection, even though microbiome microbial abundance data is not really high dimensional typically. And not as high dimensional as genomics, for example, but still they recommend using feature pre-selection. And again, the same approach, splitting your data set into training and testing, pre-selecting features and training your model. So by training their model, they mean something else. I mean omics integration. This is where I train the model. But otherwise, it's nice to see that people kind of think in this direction as well. So meaning that we are doing probably the right thing. So now I'm moving to Diablo algorithm, which is something I would really recommend you as a first step, first integrative model to use, or first choice, something you want to consider as a first step before moving into something non-linear. Diablo is simple, it's very interpretable, it's fast, and it's linear and supervised omics integration. It was developed by, I think I need to do it full screen for better visibility. It was developed by Kiman Lekao in her lab from the University of Melbourne, Australia, and it's a part of Mixomics package, which I highly recommend. It's a very, very nicely written software. So Kiman is a statistician, and all her papers and her R packages, they're very, very statistically solid and very nicely written. So it's a very robust statistical analysis. It provides a very nice visualization. So the Mixomics and Diablo, they provide very nice visualization, which is important. It has a very good documentation, and you can always ask questions. You will get a lot of support in the community. The one disadvantage of Diablo and Mixomics is that it's written in pure R, so it doesn't have C++ sort of core, and it's very slow. That's why it's very slow. And I know that Kiman was sort of thinking about rewriting the engine, at least in C++, to make it faster, because to do robust statistical analysis, you need to do a lot of resampling, which is computationally very expensive. Even though there is a lot of multi-threading implemented in Mixomics, but it's still slow. And later, when I'm going to demonstrate how Diablo works, you will see that it takes some time. And the more high-dimensional data you have and more omics you put together, the slower your integrative analysis becomes. That's probably the only disadvantage I see. Otherwise, it's a wonderful software, and I highly recommend you to take a look and use it for your multi-omics analysis. So it's based, so Diablo is an extension of PLS. Last time we talked about PLS, and briefly, it's just a very interesting way to go from the figure on the left to the figure on the right. So basically, it's a way to select features that can separate classes of your samples. So if you here on the PCA plot, you don't see males and females separated in terms of skeletal muscle gene expression. You suddenly start seeing them separated when you select appropriate features. So here PCA is based on all features, but if you manage to select the most informative ones, you suddenly start seeing your classes of samples well separated. So this is what PLS does. What Diablo does, it actually selects features from multiple omics. So here it's an example of just gene expression, one omic. But imagine that you do similar trick, you apply it for multiple omics. But as an outcome, you still want to separate two or more groups of samples. And Diablo is what I call batch effect correction. But when you work within one omic, so here you have p dimension fixed, and you put samples from different batches together. But otherwise, they both are based, both Mint and Diablo are based on PLS approach. It just the difference between Diablo and Mint is it's a horizontal versus vertical integration. But here I'm talking about this type of integration. As I said, I'm not covering integration across samples within one omic, which I call batch effect correction rather than omics integration. And so basically what PLS does, any PLS based approach, what it does, it tries to maximize covariance or correlation because correlation coefficient is just normalized covariance. It tries to maximize covariance or correlation between two types of data. So basically it tries to select features that provide the largest correlation between different data types. That's why it's integration. And by beta, I mean parameters of the model. So this is kind of, this is a model, and the model has parameters. And you find optimal parameters of the model that provide the greatest correlation or covariance between your different data types. This is what PLS does. So why is usually, if you are talking about supervised, so here we are talking about supervised integration. So why is an outcome? Why is typically it's a maybe disease status. It's a categorical, typically it's a categorical variable, healthy, sick, healthy, sick. And the X is actually your omic. It was N and P dimensions. So this is like a standard PLS. But imagine now that instead of one omic X, you use multiple omics, X1, X2, etc. And this is how Diablo works. So basically, you instead of using one omic, you use blocks of different omics. But otherwise, you use the same approach of maximizing pairwise covariance between all your omics and the outcome and the variable of interest, the phenotype of interest Y. And this is what is actually expressed in this equation. It looks a little bit complicated at first glance, but if you sort of think a little bit more about this equation, so what you see here is maximizing of the sum of pairwise covariances between your omics. So you simply compute pairwise covariances between X1, X2, etc., XQ, and Y. So you compute a sum of pairwise covariances, and you weigh them somehow. So here there is a prefactor C sub ij. That's a prefactor that kind of uses some prior information of how strongly you think X1 and, for example, X2 covary with each other. So you probably, so there are other ways. For example, MOFA is a good tool to estimate how your different omics are related, to have some idea whether they are strongly connected or weakly connected. And this information should be put as a prior information within this coefficient C in front of covariance matrix. But otherwise, what you see within this equation for covariance matrix, it's actually your raw omics X, X1, X2, etc., including Y. It's also treated as a separate omic. And A is a loading vector. So if you sort of familiar with PCA, definition of principal component is a projection of your data into a loading vector. So this loading vector comes from PCA. So you basically what you do here you compute first or leading principal components between your different data types and correlate them. So it's another way to view PLS or Diablo integration. It's a kind of, you compute principal components from separate omics and you correlate them. And in this way, you select on the fly, you select features that provide the largest sum of pairwise correlations between leading principal components. So now it's becoming really complex, maybe. So indeed, you need a little bit time to digest it, but it's actually not as difficult. So basically, if you sort of read the paper and think a little bit, I think it's becoming more and more clear the more you look at this equation and think about what exactly you're doing when you're applying Diablo.
Nikolay, so
yes.
Can this methodology be used with single cell data?
Very good question. So basically, yes, it can be. And if you're familiar with Seurat, for example, if you're doing single cell omics analysis, you're definitely familiar with Seurat pipeline. It's one of the leading workflows for single cell analysis. So they have a method of integration across omics, which is called CCA. CCA is a canonical correlation analysis, and I'm mentioning this analysis here. So CCA and PLS, it's basically the same thing. So the canonical correlation analysis and partial least square regression, they are conceptually very similar. So that's why I use them as synonyms. But if you are using Seurat pipeline, they don't call it PLS, they don't call it Diablo, they don't call it, PLS integration, but they kind of use a different terminology. But essentially what they're doing, they're doing this trick. When they do integration, they actually maximize correlation or covariance between different omics. But then in addition, on the top of that, they also use a technique which is called dynamic time warping. It's another beautiful technique for aligning time series data. So basically what they show in Seurat paper for single cell omics integration, they show that CCA alone or PLS alone does not lead to sort of to a good outcome, to what they want to see in single cell area. As I mentioned, they want to discover new cell clusters. Usually, that's one of major goals of single cell analysis. And CCA alone and PLS alone does not lead to this. But when applying dynamic time warping, which basically it's kind of it's a way to align PLS components from PLS analysis. They use sort of an alignment of PLS components on the top. But otherwise, what they're using is PLS plus dynamic time warping. So basically, short answer to a question: yes, you can use PLS for single cell analysis. It's called CCA there in the single cell area, but it's not simply CCA. It's CCA plus dynamic time warping. More questions? Or we can take them later. Okay. Right. So I need to move to the practical part. To finish with Diablo, so I'm going to demonstrate how Diablo works using 4 omics. It's samples from chronic lymphocytic leukemia cells. So it was blood samples from patients, and they used primary tumor cells and treated them with drugs in this paper from 2018. And they also did produce a few omics, a few different molecular information omics, exome sequencing they used, RNA sequencing, and DNA mutilation on these primary tumor cells. And so in the R Markdown notebook that I'm going to demonstrate a little bit later, we are going to integrate those three omics, mutations from exome sequencing data, RNA-seq, and DNA mutilation. And there will be also a drug response, some quantity for drug response on some drug treatment. So there will be four omics to integrate, and this will be a result of integration. So basically, as I said, we want to so as a phenotype of interest here, I used again gender as a very simple toy example. So I wanted to see whether I can use those omics to separate males and females. Of course, biologically more interesting would be to separate different cell types, but this information was not available in this benchmark data sets. That's why I had to stick to gender. That was like the only basically the only available phenotype of interest. And after integration, after we apply Diablo, so I will show how to apply Diablo to this multi-omics data set. So after integration, you can display your 200 samples. So this was done actually for 200 samples. So this is a result. What you see here, it's not just PCA done on separately gene expression or mutilation. So this is actually the result of integration. So features used for this visualization, they were selected through data integration. This was a picture for separate omics, and if you superimpose all these four figures, four-dimension reduction plots, if you put them on the top of each other, if you average across those four figures, this is a consensus integration plot that you get. So you really can see that you can split the orange and the blue samples, males and females, using omics integration. And here you have a lot of arrows, and it's called arrow plot. I will explain what it means, but basically it's a way to visualize both centroids of the samples across all omics, so an average position of each sample across all omics, and positions of samples within individual omics. That's why it looks a little bit complex here. You have too many arrows. But I hope still you can see that this integrative approach is capable of discriminating between two groups of samples. And I will show technical details, how to run it technically in a few minutes. So now how we evaluate whether our Diablo integration was successful. So what you essentially do when you train machine learning algorithm, you learn this hyperplane that separates two groups of samples in some latent space. So the y and x axis here, this is some latent artificial, some weird space where you project your data, and in this weird space you can linearly separate these two groups of samples. This is what essentially any machine learning classifier tries to do. It tries to find this space where groups, classes of samples are easily separable. And now when you so the the empty circles, they are actually trained data. And now when you evaluate your model on a test data, what you do, you project your test data. So the filled circles are test samples that were not used for training. And now you project your test data, your filled circles, and see whether they end up on the right or on the left side from this hyperplane. This is what was learned by Diablo classifier or PLS classifier or any other machine learning classifier. So it learns this hyperplane, the boundary between classes in some strange space. And sometimes you see that this black filled circle, it was supposed to be here on the left side from this hyperplane, but it was misclassified. So our model was not ideal. So this sample was misclassified. So our model predicts that this black sample actually belongs to the red class. I don't remember whether it was probably female. So it predicts that this male sample was actually female, which was wrong. But otherwise, yeah, you can see that there is a pretty good agreement between trained data. So the red and black empty circles and the filled ones. So you achieve a good accuracy on the test data set when you evaluate your integrative model. So Diablo has a very nice visualization. It provides plenty of interesting ways to see connections between features across omics. So, for example, here it's sort of networks. As a result of data integration, you can see connections between features across omics. So different colors correspond to different omics. So maybe some features from blue omic are connected with features from red omic, etc. Or features from blue omic can be also connected with some features from orange omic. So it's pairwise correlation networks that you can immediately see across omics, which is nice. And it has this correlation and circus plots. If you're not familiar with correlation plots, it's a way to visualize features from different omics. Different colors here, they correspond to different omics: expression, mutation, mutilation, and drug response, and treatment by different drugs. And if these features are located on the poles of this correlation circle plot, it means they should be correlated. So basically, all these features that group together on the poles, they're strongly correlated. And features on the opposite poles, they're correlated with the opposite sign, they're anti-correlated.
Nikolay, so
yes.
we have two questions. The first one, the features we are talking about all over, are the genes majorly right, or what more features can be used to do this kind of studies?
So can you repeat? I'm not sure that I understood.
Yeah, maybe it's me. So the features we are talking about all over are the genes majorly right, or what more features can be used to do this kind of studies?
Yeah, mutilation CpG sites. So here in this example, so it's features from gene expression layer are genes. From mutation, it's some genetic variation sites. Mutilation, it's CpG sites. It's mutilated, unmutilated CpG sites. And drug, I think it was it just some codes for different drugs used and some quantification for each sample. There is some level of response, how well this sample was responding on a drug. So basically, it's totally different types of information, but we still can put them together and see connections, see links between those different features, genes and CpG sites, for example.
Thank you. So the other question, can you use this approach also for the integration of omics on the same type of application, let's say gene expression, with different platforms, let's say RNA-seq and microarray?
Yes, so this is better to do with MINT algorithm. This is exactly the batch effect correction. So if you happen to do both RNA-seq and microarray on the same samples, you can on different samples. Yeah, so basically, I'm just trying to understand the question. So the way I understand the question is that the person is asking whether you can put RNA-seq and microarray. They're supposed to be done for the same genes, but for different samples. So you have two batches. One group of samples was done with RNA sequencing, and then another group of samples was done with microarray. But the genes are the same. They're supposed to be the same, or at least overlapping to a large extent. And now you want to put RNA-seq and microarray together. So I would do it through MINT. So for me, it's like two batches. They're measured with different technologies. If you are talking about maybe a single cell RNA sequence, and it can be 10x and SmartSeq 2. So you're measuring, you're quantifying gene expression levels for the same genes, but for different cells. And the same with RNA-seq and microarray. So you're kind of measuring gene expression information for the same genes, because you're working within one omic. It's a transcriptomic, but you're using different samples. And if you want to put RNA-seq and a microarray together, I would use MINT. And I know in the MINT paper, they even considered this example. So they actually, initially, I know that in mixomics, they were talking a lot about integrating RNA-seq and microarray together through MINT. But it's not something I'm covering here, because it's a batch effect correction, and I'm talking about like putting different omics together, not samples within one omic.
Thank you.
More questions? Okay, if not, then I will move further. So I think I'm done with Diablo, and I'm going now to open RStudio and demonstrate how Diablo works. But since we are talking about supervised omics integration, I thought I would like to briefly talk about nonlinear technique. So
even we had a question about that.
Okay, nice. So
yeah.
Diablo was an example of linear supervised analysis, and I wanted to show just 2-3 slides about neural networks for omics integration as an example of non-linear supervised analysis. So, but maybe you can take the question.
Yeah, so what is the difference between the linear and the non-linear omics integration? Is it related to the number of samples or the type of features, quantitative or qualitative?
It's related to the number of samples. So basically, linear analysis, it has a strong assumption that your, for example, talking about PLS, let me open it. Yes, so for example, you're looking into linear relationships between Y and X. So Y is a disease status, for example, and then you assume that this disease status is linearly connected with gene expression, for example, from X, which is a strong assumption. Maybe yes, maybe it is linear, maybe not. And ideally, you would like to test everything in non-linear way. Ideally, a non-linear integration or analysis framework is more general. However, since we usually don't have a lot of samples, we can't really always use non-linear analysis, because non-linear analysis involves more fitting parameters. So the more fitting parameters you have, the more you're prone to overfitting and a lack of generalizability of your model. So that's a danger. So that's why you should not immediately jump on non-linear analysis like neural networks. You should not, basically, whatever analysis you have, and if you're wondering whether I should do this analysis with neural network or with linear regression, I would always start with linear regression in order to be on a safe side, in order to be sure that you're probably doing your best in order not to overfit. And then if you are sort of, if you have a lot of samples, then yes, you might want to try something else. You might want to try non-linear analysis, such as neural network, which I'm going to very briefly talk about, and then open RStudio and do the practical session. So deep learning is or integration with neural networks is maybe the most flexible and most general way to do omics integration. Very intuitively clear what you are doing. Even though, as I said, these nonlinear types of analysis, they are prone to overfitting. That's why you probably have to think twice before running it. So briefly, if you're not familiar with neural networks, what they're doing, you can think about a neural network as a graph with weights. So basically, the nodes of the graph, they're connected with edges, and each edge has a weight. And here on the input layer, you provide your data. And in the output layer, you have a readout. You have your phenotype of interest, for example, which you would like to predict. And what you are doing in the hidden layers, in these nodes in the middle of the graph, you take your data, and maybe your data is a mutilation. It has this binary type 1, 0, 1, and you multiply them by some coefficients. I'll come back in a second to these coefficients. So you multiply this data by some coefficients, and then you pass this result through some nonlinear function. And then you get some output, which is your prediction, which is a prediction of your phenotype of interest. And then you compare your prediction, your predicted label with a true label. Because our prediction, it has some error, and you compute this error. And using this error, you refit your parameters. So these coefficients, they're actually the parameters of the network you fit. You optimize them. You find optimal values of these weights of the edges of your graph that provide the lowest error of prediction. So this loss function, so the deviation of your predicted phenotype from true phenotype is called loss function. It can be, for example, quadratic loss function. You use Euclidean distance to measure how similar your prediction is to your true values of phenotype. And it has typically this very complex pattern as a function of your fitting parameters. So this loss function is a function of fitting parameters, these weights of the edges. Those are So 0.6, 0.4, 0.2, those are the weights of the edges. Those are the coefficients you optimize. And if you plot this loss function as a function of the coefficients, it has this complex non-convex. In mathematics, they say non-convex shape, and you are trying to use gradient descent to actually reach to the global minimum, ideally, or at least to a local minimum of this loss function. So that's very briefly about neural network. It's a completely different topic, and it's a topic for a course, actually, how neural networks work. So it turns out that this framework is very flexible. So this type of analysis that you are doing here, predicting, so you have a function that maps your input to your output in this strange way. But it turns out that this framework is very flexible and basically fits any type of data, any type of analysis. And there is a thing which is called a universal approximation theorem. It's proven mathematically that a neural network with just one hidden layer can approximate any function, basically, with very mild assumptions on the activation function. So this function that accepts this linear combination is called activation function. So basically, in mathematics, they prove that this neural network, why it is beautiful, is because it can fit any, it can learn any profile, any behavior of any function. And this is what I demonstrate here. So basically, if you simulate, for example, this strange dependence y versus x, and train a neural network to sort of recognize this pattern, then if you use one hidden neuron, it's a little bit underfitting. Two hidden neurons, it's probably good fit. 4 hidden neurons, and I'm increasing the number of neurons here in this hidden layer. So I'm increasing the number of fitting parameters, basically. So you see that you are getting a better and better fit. And maybe 16 hidden neurons with a lot of fitting parameters. This is where you start overfitting. You see that here you start fitting the noise. Basically, this fit was good enough. So you don't have to increase your number of parameters. And moreover, if you do it, you start fitting some spurious behavior, which is a noise. You start fitting noise. This is what is called overfitting. So basically, this is another demonstration that why you should not immediately do nonlinear analysis, because nonlinear analysis typically means a lot of fitting parameters, and then you might end up with this situation. So you probably will be fine with just a few fitting parameters for this particular example. But here I just wanted to demonstrate why neural network approach is so flexible. It can basically learn any functional behavior of any data type. And in theory, they say that also neural networks and deep learning approach is becoming superior when the amount of data grows. So this is this schematic picture shows that basically neural networks they typically outperforming traditional statistical algorithms and also traditional machine learning methods such as random forest, for example, when the amount of the data grows. So they're really well suited for analysis of large amounts of data. And we actually do have large amounts of data in life sciences. So if you're doing computational biology, it's not straightforward to use deep learning everywhere. For example, for genomics, it's hard to do it. But in single cell analysis, this is where this area is a paradise for deep learning, because you really, as I mentioned already, you really working with millions of cells. So it's quite common now, and it started maybe 2-3 years ago, when data sets with millions of cells became available. And now people are working with 4 or 5 million cells. It's quite common. So this is where you, this is an example where you really want to, if you do integrative analysis, you really would like to do it in non-linear way, because linear way will not be good enough. Maybe you will be here, you will be underusing your data. You can't, with a linear model, maybe you will not be able to capture all the patterns in your data. So you really, you do want, with a lot of data, you do want to increase your number of fitting parameters in order to be sure that you don't miss any interesting pattern. And so basically, if you want to do integration with neural networks, then single cell omics is something for you to test your ideas and your models. And this is basically how you would use a neural network for supervised data integration. So if you have 4 omics, you just put them as 4 separate layers, input layers. And on the output layer, you put your phenotype of interest, for example, disease status. And then you kind of, what you do here, you merge, you concatenate all these layers, but not immediately, you don't concatenate input layers, but you first, you transform your data in some common space. So a neural network belongs to the class of integrative algorithms that first convert your different data types into some latent common space where your different data types, they forget about technological differences and they can be simply merged in this common latent space. This is what you're doing here. And on the fly, you can do a lot of beautiful tricks, such as regularization. You can do feature pre-selection on the fly, which you can do with lasso. Within neural networks, you can also use lasso, but there is a like a native regularization technique in neural network, which is called dropout. So what dropout does, it basically drops 1 or 2 or a fraction of nodes randomly, and the neural network has to survive without these nodes. In this way, it learns only the most informative nodes. It learns where actually how the information passes and what nodes can be omitted and what nodes are really important to keep in this graph. This is what dropout does. It's a beautiful regularization technique, but conceptually it's the same thing as doing lasso, or basically it's the same. It's called regularization, that you are trying to figure out most informative features and get rid of the garbage, get rid of the redundant features. Yes, so you can do this regularization on the fly. You can convert your omics into some non-parametric space, which might be like sort of PCA space. For example, if you're using linear activation functions here, for example, if your activation function is linear here, then your neural network is equivalent to principal component analysis. But it does not have to be linear. So if your activation function is non-linear, then you do some non-linear dimension reduction. So you convert your omics into some non-linear latent space, and in this space you can concatenate them. And here you predict your readout, your phenotype of interest, and you update your weights of the graph. By the way, I forgot to say that the way these weights are updated is called backpropagation, because you compute this error, and then you recalculate the weights of the edges backwards. So you first update these weights and then update those weights. So you're moving from the end of the network to the beginning of the network. That's why it's called backpropagation.
Nickolay, we have 30 minutes remaining.
Yes, I'm moving to the practical session now. So that was actually the last slide. Thank you.
We will answer the question at the end of the session. We have many questions, but it's better to move now to the practical.
Yes, so then I'm opening my RStudio, and I'm making it as
can you zoom
large as possible. Is it okay like this?
Yes.
Okay, excellent. So this notebook is available on the GitHub page of our course. I posted the link. If you have troubles accessing the notebook, let me know, and I will sort of I will post it again. So here, what we are doing in this notebook, we are analyzing. So we are doing integration of mutation data, gene expression data, mutilation, and drug response from the same samples. So we are using the CLL data set. And as a first step, we are starting with exploratory data analysis and also cleaning and understanding our data. So I start with gene expression. So I read the gene expression information. We can visualize it, how it looks like. Yes, so the columns here are genes with ensemble IDs, and the rows are the samples. And since I know that some of the omics, they have missing data, so I immediately apply imputation, median imputation. So what I do here in this line, I basically take, for each gene, I'm looking whether there are missing values in the column. And if there are missing values, then I'm using a median of this gene expression across all samples as a sort of predicted gene expression for the missing sample. So it's called median imputation. You impute missing value with a median gene expression across samples. So you can sort of run this as just one line, and so basically I don't think there were missing values in gene expression, but I'm always kind of using this imputation step in order to ensure that I don't have to manually check every time whether there are missing values or not. And now there are mutations, and this is how mutation data looks like. Again, the columns are your genetic variation markers, and the samples are the same. So please, yes, please note that the samples are the same, but the features are different. So we are integrating across omics, not across samples. And now mutilation data. This is quite a high dimensional data. So you can check dimensions here. So for mutation, for mutilation data, we have 4,248 CpG sites. So the columns are CpG sites, and the samples are again the same across omics, across the 4 omics. And there are 200 samples. So basically, if you look at the dimensions, so mutation data frame has 200 samples, and mutilation data frame has 200 samples. But the features, the p dimension varies. They are different for different omics. And finally, the drug. So it looks like this. So again, it's some drug code, which was, so these samples were treated with different drugs, and this is some quantification. I'm not sure how exactly they quantified the response of the sample on a drug treatment. And finally, this is the only phenotypes that were available from this benchmark data set. And unfortunately, all of them are CLL samples, so there are no other samples. That would be, of course, very cool to use two classes, CLL and maybe ALL samples, and to find a separation between those different samples through omics integration. But unfortunately, the only phenotype which was available was gender. So that's why I'm using it as a readout, as something to predict. And now, so once we kind of read our data, we are going now to split our data set into train and test subsets. So since there are 200 samples in total, so I'm saying that I will reserve 60 samples for testing. So it's approximately 30% of samples will be used for testing, and 140 samples will be used for training. And so for simplicity, I select 24 females and 36 males. So the 60 samples will be 24 females and 36 males, which is approximately like 40%. We know from 200 samples, if you look at the percentage of females, it will be 40%. And we will use exactly the same ratio, the fixed ratio between numbers of females and males. We will use also for the test sample, for the test subset. So we will select randomly 24 females and 36 males, and those are the female and male samples that will be reserved for testing. And here I am building gene expression, mutilation, mutation, and drug response data set with those IDs for the female samples and male samples that were reserved for testing. So expression test underscore test and mutilation underscore test, etc. Those will be the test data sets that I will be. I will train my integrative model and use those test data sets for predicting the gender at the very end. And this will be my phenotype, test phenotype. So
Nickolay, so there's a question: what are these values in the drug data set? What do they mean exactly?
Yeah, so this I can't answer, unfortunately. It's I only can say that this information can be found in this paper. So unfortunately, don't remember it was a long time ago since I read this. I'm not sure how they quantified the drug response, but it was some sort of some quantification. So I'm sorry.
Can you put this reference in the chat? So maybe.
Sure, absolutely. Yeah, that's a good suggestion. Yeah.
Thank you.
So if I want to put it into the chat, I have to stop sharing probably. Yeah, I put it here. And now I share my screen again. Right. So so until now, I was building test data set, and now what I'm doing, I'm building train data set by including samples which are all the rest samples but the test samples. So you exclude test samples and keep the rest for training. And if you look at the dimensions, it will be 140 samples. And by the way, for the expression data set, there were 5,000 genes, mutilation, mutations, drug response. So, and again, this is a summary. If you look at the Y variable as the gender variable, it's 140 samples, 85 males, 55 females. So I'm going to use this information for training and hopefully make a correct gender prediction on the reserved 60 samples. So here in this notebook, I'm doing some additional filtering of mutation data set, because if you look at if you work with genetic variation data, so you know that it typically so in this case it has only two values in this matrix, and it's 0 and 1. So it's actually kind of minor allele present or absent. And basically, this is a binary data type, and binary data, they usually have very little variation. So basically, since it's only 0 and 1, there is not enough sort of information to vary across samples. And that's why here I'm using this near zero var function that removes genetic variants that do not vary across samples, that are zeros everywhere, for example, or only just a few ones across samples. So if you're familiar with genetic variation data type, it's basically equivalent to excluding rare variants and keeping only common variants, variants that are present in 5% of samples at least. So I'm demanding here that I want to see my minor allele present in at least 5% of all samples. So basically here I'm removing a lot of genetic variants, and at the end there will be only 11 genetic variants kept. At the beginning, I think it was hundreds of genetic variants, but since it's a binary data type, so you have to prune it somehow, because otherwise PLS will have troubles, and many other statistical algorithms will have troubles doing multivariate analysis on this binary data set. Binary and categorical data sets, they are unfortunately difficult to analyze, so you need to filter them quite a lot before proceeding with any analysis, not only integrative analysis. And yes, so here, yeah, I think I excluded for some reason. I excluded all my columns. This is strange. Yeah, I will read it again. And now I prune it. So now it works.
So what's the meaning of pruning the data?
Yes, so what we are doing now, we are trying to exclude genetic variants that are too rare. So the minor allele, if you're familiar with minor allele frequency concept, so the minor allele is present in just a few samples, but this is kind of very dangerous to analyze, because you can't establish a good association with a minor allele count, because there are too few minor alleles. So there is not enough statistical evidence to analyze these genetic variants, and these genetic variants I would like to exclude. Yes, so something went wrong apparently. Yeah, so basically if you run the notebook, maybe I should switch to, in order not to try to figure out the error, which was something strange happened with, I was running these codes a few times just to test that they're working. So I will switch to the HTML version of this notebook in order to make sure that that you get sort of right information instead of just spending time on looking for bugs. That's why these HTML files are very handy. So here, eventually, what we are doing, we build train and test subsets, and we proceed. So here we pruned mutation data sets. So we are preparing our data for integration. So we need to exclude non-informative features from a mutation data set. And also, I'm going now to do some feature pre-selection on the expression data set, because expression data set included 5,000 genes. This is a high-dimensional data set with only 200 samples. So I'm going to use lasso that I explained last time and use this gender as a phenotype of interest in order to first do cross-validation and find optimal lambda, optimal penalty for lasso that I explained last time. Through cross-validation, we are looking for optimal lambda parameter. And then use this optimal lambda for selecting most informative genes. And instead of 5,000 genes, we end up with only 32, I think, genes. So Lasso selected, and Lasso said that out of 5,000 genes, there are only 32 informative genes. So the rest can be removed. This is a dramatic dimensionality reduction. So we went from 5,000 genes to 32 genes. And so basically, this is a list of these genes, and I'm going to keep them for integration. So what we are doing here, we're doing this feature pre-selection step before running integration. And I'm going to do the same thing for mutilation data, because mutilation data was it contained also like 4,200 CPG sites, so not all of them are informative. We need to reduce the number of dimensions in order to overcome the curse of dimensionality. Again, I am doing it through lasso. I'm looking for optimal lambda, optimal penalty, and I end up with only 28 CPG sites, so out of 4,000. So luckily we can remove most of the CPG sites. So now we reduce dimensions in the mutation data, in gene expression data, and in mutilation data. So we can proceed now with integration. So we dramatically reduced dimensions in each individual omic. So now we actually proceed with the integrative analysis. In the first step of Diablo integration, we create this list of our omics: expression, mutation, mutilation, and drug response omics. So here we can also display the dimensions. So that's important to see that there are 140 samples in all 4 of them, but the number of features you see there, it was reduced dramatically. So from 5,000 genes, we went down to 32 genes and 10 mutations only, and 28 CPG sites. So now we have kind of, if you sum up 32 plus 10 plus 28 plus 310, it will be still greater than 140, but not much greater. Sort of we are trying to avoid the curse of dimensionality as good as we can. That's why we did this feature pre-selection. And now we define our design matrix, the so-called design matrix for Diablo integration. which is basically, if you remember the C coefficients in front of pairwise covariance matrices in the equation, in the Diablo equation here. So basically, the C coefficients, those are defined in the design matrix, and this is some prior information. You can sort of here I simply say that all my omics are correlated. I put coefficient 1 for any pair of omic because I don't have any other prior information, but you can do it in a more clever way with MORPHA. You can quantify how well actually related, genome-wide related pairs of omics are. Next, once you define this design matrix, next you're doing what you're doing here. You're doing you're actually trying to figure out. You're performing first iteration of integration with PLS. Here you provide all your 4 omics as data. You remember the data was a list of our 4 omics. So you provide here all 4 omics and the phenotype of interest as a Y variable. And now you try to figure out how many PLS components you would like to keep in your analysis for accurate prediction. And this is a hyperparameter. The number of PLS components is a hyperparameter, which can be optimized through cross-validation. And this is what this perf function in Diablo does. So it sort of it tries to plot an error, get this plot of error of your prediction as a function of number of PLS components to keep in the analysis. And you remember when you do cross-validation, you always plot, you always kind of estimate an error of prediction as a function of a hyperparameter. In this case, a hyperparameter is a number of PLS components to keep for further downstream analysis. And the error seems to be dropping dramatically from 1 PLS component to 2, and then it kind of saturates. It doesn't decrease much anymore. So we can keep only 2 PLS components. That should be enough for a good prediction. And finally, we are doing another tuning of a Diablo algorithm with lasso. Now we are doing tuning this algorithm with lasso, because we are saying that despite we did some feature pre-selection, so we reduced dimensions, but we can still do another round of feature pre-selection on the fly while doing integration. And this is what we are doing here. We are trying to even further reduce the number of dimensions while doing integration. And we end up with these numbers of selected features. So we went down to just a few features. So we are narrowing down more and more the number of features, most informative features through integration. And finally, we are arriving to this plot, which is sort of it's using. So in order to produce this plot, you actually you are keeping those features that were selected in several ways. Actually, you first did feature pre-selection and then another feature selection when performing integration. And if you want to visualize what kind of features, you can of course see that those were the genes, for example, that were contributing the most to the PLS component 1. And using only those most informative features from different omics, you can see that your samples, females and male samples, become somewhat separated, at least for expression, maybe for mutilation a little bit, but not astonishingly separated, unfortunately. And you can also visualize the importances of expression markers, mutilation and mutation markers to check how they contribute, in which direction they contribute. So this is what you can get from Mixomics. It has a very nice visualization in this sense. And finally, so basically the final result, absolutely the central result of integration, is this arrow plot, which sort of gives you a visualization of your samples, males and females, in a common space, in the common latent space. And it looks like those two groups of samples can be linearly separated. So there is some, you can see that there is some separation between orange and blue samples. And again, you can interpret your model. So Diablo and Mixomics, they have a nice interpretation plots. You can check how these different selected features, they what kind of interplay they have, how they are linked through different plots. From this circus plot, where it's just a connection between different features across omics. So here, what you have here, there are features, feature names in different omics: expression, mutilation, mutation data, and you can visualize how features across omics are linked. And also you can do this networks again. In order to visualize links of features across omics. And you can also do prediction. Now you use the test data set. You prepare a new test data set with 60 reserved samples, and you use your trained Diablo model for predicting on the new data set. And you can sort of display so-called confusion matrix, where you have a true label, male or female, and you also have a prediction from different components, PLS components for different layers. And you can use sort of majority voting to see how much each omic contributes to the prediction, and also compute accuracy. So in this case, the accuracy of prediction was not fantastic; it was 61%. But this is a good, I think it's a good benchmark data set where you can sort of check and try to understand Diablo. And I hope you, despite I'm running out of time, I hope you at least will remember this method Diablo, and might want to have a look and use it for your research. So I think I'm done. Thank you very much. I'm sorry about this 11 minutes delay.
No, we have still until 15, so you have 4 minutes more.
4 minutes for answering questions.
Yeah, if you want to say more things, go ahead, please.
No, I'm done. I will be happy to answer questions, and if you don't manage to answer all questions, I'll be happy to answer them offline. So guys, please don't hesitate to drop me a line, and I will be happy to answer your questions.
Thank you very much, Nikolay.
So, Hedia, can you read a few questions?
Because I hear some like background noise. So we don't have to do any pre-processing with the drug data?
Yes, so the drug data, indeed, I didn't do feature pre-selection for the drug layer because it was not really high dimensional. It was only 300 drugs tested on these tumor cells. That's why I didn't want to reduce dimensions any further. But if you look at mutilation or gene expression data, they were high dimensional. That's why I did feature pre-selection for those layers, but not for the drug layer.
Thank you. So there's another question. If you have multiple data sets from cell lines, and for each of these samples, you perform single cell sequencing, whole genome sequencing, proteomics, would you use Diablo or MINT? Thank you very much.
So MINT is an algorithm for one omic. So it's not really omics integration. It can be viewed as data integration because you do combine different data sets. but MINT works for one omic with measurements for different samples, different biological replicates. But Diablo is integration across omics. So if you have transcriptomics, proteomics, metabolomics, genomics data for the same biological samples, then you use Diablo.
Thank you. So the other question is about the activation function. So is there any recommendation for the choice of activation function in the hidden layer for single cell, for example, sigmoid or ReLU versus ReLU?
Yeah, so I know that deep learning people, they kind of use ReLU all the time. So I always start with linear activation functions because I want to kind of, when I'm sort of doing development, when I'm developing my network, I always start with linear activation functions because they are more like easier to understand the outcome. ReLU actually works very well for image analysis, and you don't always do image analysis. If you don't do image analysis, you probably you should think twice before using ReLU. So please remember that this fashion for using ReLU, it came from image analysis in CNN, convolutional neural networks. So you don't have to use ReLU all the time. So sigmoid, yes, that would be like a first upgrade, first non-linear upgrade with respect to linear activation function. So if you want to replace your linear activation function with a non-linear activation function, I would use, yes, sigmoid or hyperbolic tangent function. And yes, ReLU and many other functions, activation functions are possible, but you kind of, it's a hyperparameter. You can always test a few activation functions and see what works best for you through cross-validation. Remember that any hyperparameter, such as number of layers, number of neurons, activation function, loss function, they can be tested through cross-validation.
Okay, so what depends the choice of the activation function of?
Yeah, so it's a good question. So it doesn't have a simple answer. So it's a little bit intuition sometimes. People have some rules of thumb that if you do NLP, for example, natural language processing, you use one type of activation function and one type of optimizer. So there are also optimizers, different types of gradient descent algorithms or modifications of gradient descent, how you find this local or global minimum of your loss function. For NLP, they use one type of optimizers and activation functions. For image analysis, they use another type of activation functions and optimizers. For tabular data, so what I was talking today about is actually tabular data. And there you, basically there are other rules of thumb. So it's a little bit, there is no like straight answer to your question. What people know is that a choice of optimizer and activation function depends on the data type, definitely, but it also kind of, you can always test a few and see what works best for you. Sometimes very unexpectedly, you end up with something unconventional, something what people don't read in literature, some functions that they don't report in literature, but something that works for your particular data type.
Thank you. So we are thinking now the last question. So are there Python packages that can do omics integration that work as good as mixomics?
Yes, good question. I'm not aware of Python analog for Diablo or mixomics. I know that there are actually a few Python packages for unsupervised omics integration, such as SNF, Similarity Network Fusion, for example, beautiful unsupervised omics integration algorithm. There is definitely a Python version for SNF, but for Diablo, I know that, well, there is a PLS there is a PLS definitely in Python, but no Diablo. So Diablo is an extension of PLS. So I'm pretty sure that you can do PLS in Python, but to do Diablo in Python, I'm actually not aware of such a package. But maybe Kim Anlikov is working on something.
Okay, so does Diablo take RNA-seq gene expression and single-cell expression also?
Yes, so you can run you can run Diablo on single cell data definitely, and I did it, and basically there are no major restrictions for this. You can do it, but single cell community they're a little bit they were for some time separated from the bulk sequencing community, and they were kind of developing their own methods, which as I mentioned, they developed a CCA plus dynamic time warping, and CCA is a PLS. So basically, I think they were very much, some of them were very much inspired by Diablo, but they kind of presented a new method which was suitable for single cell, although they could have used Diablo for single cell. Yeah, but it's just a question of, you know, different traditions. People from different communities, they unfortunately don't talk to each other that much, and they kind of rediscover things and reinvent some old algorithms.
Thank you. So the last question, what is the final result in the example shown during the workshop? What are gene expression, mutation, mutilation patterns, and drugs that the model used to separate men and women? Where can we see this result?
Yeah, so right. So basically the main result of the integration, as I said, was this visualization. So, well, you don't see it because I'm not sharing my screen. So the main result of the integration was this arrow plot. So it's a visualization of male and female samples in the common space. So this is a result of integration. This is a visualization of the samples after integration has been done. But you can always check the loadings of this plot. And so in the notebook, all these features, selected features, they're actually written there. You can check what kind of features underline this plot. It's straightforward to see what kind of genes and mutations are responsible, are driving these separations on the consensus plot.
Thank you. There's another question, but we really ran short of time. So thank you very much, Nickolay. Thank you everyone for joining our R4Bioinfo. And please stay tuned about our next workshops. Thank you.
Thank you very much, guys.
Thank you. Bye-bye.
Thank you.
Maybe you can run like unsupervised for next time for us.
Absolutely, I would be happy. Yeah, you asked me to show supervised methods. That's why I was concentrating.
Yeah. Thank you very much.
I will be very happy to talk about UMAP integration. And if you're interested in single cell analysis, there are plenty of unsupervised tools. And since single cell is a trending thing, I assume many people would be interested in doing unsupervised.
Because there were many questions today about that.
Indeed.
Thank you. Thank you very much. Maybe we'll have unsupervised session coming soon. Thank you. Thank you very much, Nickolay.
Thanks a lot, everyone. Thank you for joining.
Bye-bye.